So in Lab 2, you're going to be talking about the microscope. Now in previous videos, Liz and I showed you the parts of the microscope, how they work, how to focus a microscope, what everything does. So in this video, what I want to show you is a little bit about what you're actually going to be seeing under the microscope. So there are 3 slides that you're going to be exploring in Lab 2. The first one is this, which is the letter E. So when you get the E, what you're going to do is put it onto this stage in the stage clips so that you can actually read the E as if it was typewritten, right? You can see it as you would expect to see an E. You're going to put it as close to the center of the light as possible, and you are going to then try to focus on it. Now, if you're lucky, when you get it in, it will look like this, but more than likely you will have to play with the coarse focus and then fine focus to get it into view. So once you get it here, you are going to get it into a picture perfect view, right? So you'll use your fine focus. You figure out which way you're going. So I'm using the fine focus right now to get this into view. And you can see that that's pretty clear. I think that's a pretty clear picture. And then what you're going to do is use the control knobs that are on the right side of the microscope to get this as close to the center of your view as possible. So this right now is under 10x. I'm sorry, this one's under 4x. This is our scanning microscope, or our scanning objective. So we're on the very lowest power, and you can see the E. However, you should notice that this doesn't look like what you would think an E does. That's because the E under the microscope is inverted. It is upside down and backwards from what we would see it on the slide. That's because of all the lenses that are in the microscope. It flips things upside down and backwards. So it's not just upside down, it's inverted, upside down and backwards. So you can still see the E, it's just that everything's backward, which means when you move the E from right to left, it actually looks like it's moving from left to right. So whenever you're moving something on the microscope stage, it actually looks backwards on the scope. So if you move it forward, it looks like it's moving backwards. If you move it left, it looks like it's moving to the right. So it takes a little bit of adjustment for those of you who haven't done this before. So I'm going to walk you through what is basically procedure 1 here in your lab book. So procedure 2.1. So we have the E in focus under 4x. Now remember that our microscopes are parfocaled and parcentered, which means once they're here, you can basically just slide them into the next objective without a whole lot of effort, which is very, very nice if you've ever used other microscopes that aren't quite this easy. So I'm going to go ahead and move this microscope from the 4x to the 10x. So right now we are on the 10x objective, and this is magnifying by 10 times, and then our oculars, which is another 4 times. So this is a 40x magnification. Now what you'll notice is the E is basically still in the center, right? And it's a little bit blurry, right? It got a little blurrier as we moved up. So you are going to want to, again, use the fine focus. So I move the fine focus one way, it gets blurrier. You move it back the other way, and it should get a little more clear. So the E now is more zoomed in. We can see it. It's still inverted. Now once you get here, you are going to put it as close to the center of your view as you can. So here we're centered. And what we can actually do here is go up to the highest magnification that we have in our lab, which is the 40x objective. So I'm going to go ahead and slide that, sliding into our 40x. Now what you've noticed here is you can still kind of see the E, right? But you can't see all of it anymore. We lose the entire picture as we zoom in, right? Because we're looking at a piece of it. You can also still see that it's really, really blurry. So what we need to do is, again, use the fine focus to get this right into picture perfect view. And here we are. So once you get here, we can see the line. So this is the middle line of the E. Now if you do choose to move your E around a little bit, you can use the stage controls to do that. You have to be super gentle as you do, but you can then move around and see the other parts of the E a little bit closer. So you can see there's an open spot on the side of this E, but you can move it around. You have to be very, very gentle, but it helps you to explore the close-up version of the object that you are looking at. So with the E, it's just part of it. It's just one small line in this case. So we lose the whole picture, but we gain more detail detail on some parts of our object. And this actually ties into one of the last experiments or procedures you do in this lab, which is about depth of field. We lose the depth of field. The other thing, I think maybe it didn't come through quite as well in this program because this program adjusts for brightness automatically a little bit, is that as we zoom in, we also lose brightness. So the light on the microscope will decrease as we zoom in. So you can play with adjustment of the light, right? You can use the iris diaphragm, which is right under the stage, to make it darker or lighter, which I'm doing right now, even though the program is not letting me as well. It like readjusts everything. That's kind of funny. On the microscope, you will not have auto adjustment. The computer is doing that for me. So I'm also going to look at procedures 3 and 4 in the next videos, looking at how we measure the diameter of our field of view, and then we'll look at the threads, which are the last one when we talk about depth of field.